This video focuses on stage 2 of photosynthesis, or the light-independent reactions. So if you remember from the light-dependent reactions that take place in the thylakoid, we got NADPH and ATP out of those processes. So now we're in the stroma, and we're going to undergo the light-independent reactions. The whole point of photosynthesis is to take the energy from the sun and turn it into something useful for the plant. So far we've taken the energy and put it into electrons and used that energy to create these NADPH and ATP molecules. Now that we're in the stroma, we also have a couple other molecules that are going to be useful. So we have carbon dioxide from the atmosphere. We also have ribulose bisphosphate, or RUBP. And we have an enzyme called Rubisco, or ribulose 1,5-bisphosphate carboxylase. So we just go with Rubisco for short. And this enzyme Rubisco is used in the first stage of the light-independent reactions. We call this first stage fixation. And carbon fixation involves converting inorganic carbon, something like carbon dioxide, to organic carbon. Now, organic carbon has carbon bonded to hydrogens. Organic carbon are things like glucose, which has carbon bonded to hydrogen. So the enzyme Rubisco is used to convert inorganic carbon dioxide into an organic carbon using RUBPH. Now these molecules combine with the help of the enzyme. They create a very unstable compound, so it immediately changes into a more stable form. And what it changes into is 3-phosphoglycerate. So you don't need to know all of this chemistry, but I do need you to know the process of how this carbon dioxide gets eventually converted into sugar. So these phosphoglycerates are more stable, and they are an organic compound. So the carbon dioxide has now been fixed from an inorganic form to an organic form. So that ends the fixation stage. In the second phase, reduction, we want to create more useful organic compounds. So we want to take what's been made so far, and we want to change it into something that can be converted into carbohydrates, which are actually useful for the plant. So this is where our NADPH and ATP come in. So the NADPH is going to contribute electrons, the ATP is going to contribute energy, and it's going to convert this organic compound into the more useful glyceraldehyde 3-phosphate, or G3P for short. Now this G3P is incredibly useful because it can be easily converted into any type of carbohydrate that the plant could need. So it could be converted into glucose for use right away. It could be converted into cellulose for short-term storage, or starch for long-term storage. So now that we've converted the energy from the sun into useful carbohydrates, or carbohydrates that we can use in the plant, photosynthesis is kind of finished. But this is a cycle, so we need to reset the cycle for this to be able to continue. So some of the G3Ps that were made in the reduction stage go on to make these carbohydrates, but some of them go on to regenerate the original reactants. So the next phase is regeneration. So in order to replenish the RUBP and continue this cycle, the regeneration phase requires G3P. So some of the G3P that was made doesn't leave the cycle, it just goes on to this regeneration phase. It also requires the energy from ATP and NADPH. So those molecules that were made in the light-dependent reactions are required again. And all of these contribute to the regeneration of the RUBP. Once we've regenerated the RUBP, more carbon dioxide can come in, and the cycle can continue to produce the G3P, which is converted then to the useful carbohydrates. So to summarize, the first stage in the light-independent reactions is fixation. Fixation takes the carbon dioxide and turns it into an organic carbon molecule, which the plant can use, and this requires the RUBP as well as the Rubisco enzyme. The second stage in this cycle is reduction. In the reduction phase, we have the formation of a stable organic compound, and then with the addition of electrons from NADPH and energy from ATP, we form the G3P, or the glyceraldehyde 3-phosphate. Now this is the compound that can then easily be converted into any carbohydrate that the plant needs. The last phase is regeneration, where we need to regenerate the RUBP that was used up in this cycle. So we need the ATP, NADPH, and the G3Ps that didn't leave the cycle to regenerate the RUBP. So this is like the reset phase for the cycle to continue. So the cycle is called the Calvin cycle, and it uses the products from the light-dependent reactions to basically take the carbon out of the carbon dioxide and put it into something that the plants can use to store their energy.